So here's the second part of notes from this chapter. Complete dominance. All of the genetic crosses we have done so far from worksheet 1 and 2 demonstrate what's called complete dominance. This is where one gene is dominant to another, which is recessive. So we have that dominant gene masking a recessive gene. Today we're going to talk about incomplete dominance and codominance. So for incomplete dominance, this occurs when no gene is dominant. Rather, when different genes appear together in a gene pair, they blend to form a new trait. So actually, they're both dominant, but they're just not dominant over each other. So our first example here, you see we have a red flower crossing with a white flower. Both red and white are dominant traits, dominant genes, and they will blend to form a heterozygous RW, and that phenotype would actually be pink, not red or white. So sometimes you have this mixture type phenotype. Another example would be a black rabbit mating with a white rabbit, which would make a gray rabbit. Both black and white could be dominant genes. So for incomplete dominance, once again, dominance could not be completed because neither gene is more dominant than the other. They are both dominant, so you blend together. Another example would be a curly-haired person mating with a straight-haired person to create a wavy-haired person. We have this genotype here. This would be our heterozygous. Or you have an example in cows where a red cow would mate with a white cow to form what's called a roan cow. Here are a few more examples. Here are our genotypes. This would be homozygous dominant curly. This would be heterozygous. And this would be homozygous dominant for straight. This would be homozygous dominant red, heterozygous, and homozygous dominant white. So here's how you would do a Punnett square in that situation. If we're looking at flower color, I'm telling you red is dominant, but white is also dominant. So if we have a red flower crossing with a white flower, here's how we would set that up. We have all capital letters here. Capital R, capital R, capital W, capital W. All of our offspring here would be heterozygous RW, and our phenotype would be a pink flower. So here's how you would set that up, and here's how you would write out those genotypes. So sometimes both genes are dominant. Neither one is dominant than the other, so we get a blending of traits for our phenotype. Here's another example for hair shape using curly and straight. So it's not exactly complete dominance like we looked at on worksheet 1 and 2. It's more like a incomplete dominance. So we would have a curly-haired person crossing with a straight-haired person. We'd be homozygous dominant curly, homozygous dominant straight. All of the offspring here would be heterozygous wavy. So all 4 offspring chances for heterozygous, and the genotype would have wavy hair. So it's a combination of straight and curly. Another type of dominance that we will talk about here would be codominance. Codominance occurs when 2 genes are both dominant or codominant to a third gene which is recessive. So if you're talking about genes that are codominant, they are equally dominant to each other. So they are equal. So instead of seeing a blending of traits, you're going to see both traits equally. For example, if you had a black rabbit and a white rabbit, instead of making a gray rabbit, with codominance, you would make a white and black spotted rabbit. Another really good example would be blood types. Blood types are controlled by 3 genes. We have gene for A, B, and O. A and B are dominant. They are codominant to each other. And O is recessive. So we're going to do some crosses demonstrating codominance here. But just to show you for blood types, because blood types are very interesting. Here are all our possible gene pairs. Here we have capital A, that's homozygous dominant A. So you'll see here, we're going to have a couple homozygous dominants, here and here. However, we have to say HDA, because if I just say, oh, homozygous dominant, you don't really know if I mean homozygous dominant for A or for B. So that's a big difference here. So we need to make sure we're saying homozygous dominant A. So what blood type would this give us? Blood type A. This AO, this is a genotype. This would be heterozygous for A. And the blood type would be A because A is dominant over O. Here we have homozygous dominant B, so 2 capital B's. That would give us blood type B. Here we have heterozygous BO. B is dominant over O, so this blood type would be type B blood. Then we have AB. Both A and B are dominant. We have 2 different letters here, so we do call that heterozygous, but we have to say het AB. So do we have A blood or B blood? Well, neither. Because A and B are codominant to each other, they are equally dominant, this would be type AB blood. You're going to see both A and B here in this situation, and you'll see them equally. Our last gene pair here would be 2 O's, and these are both recessive, so this is the only time we could actually use homozygous recessive here. And there's nothing dominant to mask anything here, so this blood type would be type O blood. So let's see how this works. Here's a blood type problem. I'm just making this up for us for practice. We have our dominant forms of blood type would be A and B, and our recessive would be O. I'm telling you that mom is AB, and I'm telling you that dad is type O. So I'm giving you their phenotypes. I'm giving you their blood types. So there's actually only one genotype for each of these. Mom would have to be AB, and dad would have to be OO, and they're both lowercase o's. So if we were to fill in this Punnett square, you would see that we would have 2 heterozygous A and 2 heterozygous for B. So that's how we would write this over here. 2 het A, 2 het B. What blood types would we get here? This would be, we'd have 50% chance, 2 out of 4, type B, and a 50% chance, 2 out of 4, that this child would be type A. Fun fact for you, this is actually my blood type here, and this is my husband's blood type. So I know if we have children, which we have a 2-year-old and one on the way, I know my kids either will have to be type A or B blood. Here's another example. What if mom and dad are both AB? Here's how we would fill that in. So interpreting it, we have 1 homozygous dominant A, 1 homozygous dominant B, and 2 heterozygous AB. Now remember, you have to tell me over here, because there are many that could be homozygous dominant or het. So you have to give me dash what it's for. Phenotype-wise, we have 1 type A, 2 type AB, and 1 type B. So 25% chance for type A blood, 50% chance for type AB blood, and another 25% chance of having B blood. You also have the RH factor on blood. This is an additional protein found in the blood of most of the population. About 85% of the population has this protein. If you have the protein, you are Rh positive. If you lack it, you are Rh negative. It is inherited through complete dominance, so just like worksheet 1 and 2. It is actually linked to rhesus monkeys, hence the Rh in rhesus, the Rh factor. And we'll simply use the symbols plus and minus instead of writing out the terms. So there are 3 different gene pairs here. You could be homozygous dominant, positive, positive. That would give us the phenotype of a positive blood type. You could have positive negative, which is heterozygous, which would give you a positive blood type because positive is dominant over negative. Or you could have negative negative, which is homozygous recessive, which is the only combination that will give you a negative blood type. So this is just like the very simple squares that we did on worksheet 1 and 2, except we're not using letters. Here we use symbols. So looking at worksheet 3. Number 1 is blood types. Telling you once again, dominant is A and B, recessive is O. Telling you that mom is het. I didn't tell you for what. And dad is het AB. Do all possible crosses, list the genotypes and phenotypes. So if mom is het, and I'm not telling you what she's het for, there are a couple combinations for her. She could be het A, het B, or het AB. We don't know. Dad is het AB, so we know exactly what he is. So how many squares do we need to do? 1, 2, and 3. So we have 3 squares that we need to fill in, which I have color-coded for you. So here I have mom being AO and dad being AB, mom being BO, dad being AB, mom being AB, and dad being AB. So once you fill those in, we do the genotypes and phenotypes for everything. It can get a little, you have to do a little more writing than what we did on worksheets 1 and 2, because for blood types, we have to indicate what blood type in our genotype. So we have, for our first box, 1 homozygous dominant A, 1 het A, 1 het B, and 1 het AB. So that'd be 2 blood types for A, 1 AB, and 1 B. Over here, we have 1 het AB genotype, 1 het A, 1 het B, and 1 homozygous dominant B. Phenotype-wise, we have 1 AB, 1 A, and 2 B. Both of these would be B. For our purple one, we have 1 homozygous dominant A, 2 het AB, 1 homozygous dominant B. Phenotype-wise, 1 type A, 2 type AB, and 1 type B. So that is number 1 on worksheet 3. Skipping to number 4, just to show you the Rh factor and how it's just like complete dominance. I'm telling you dominant is positive, recessive is negative, mom is positive, dad is negative. So mom could be positive, positive, or positive, negative. That's homozygous dominant. This is homozygous recessive. Dad is negative, so he could only be homozygous recessive, negative, negative. So we have to do 2 squares here. Here they are. Fill everything in with the symbols. For our first square, all 4 offspring chances are het, and all 4 would also show the positive blood type. Here we have 2 het and 2 HR. So blood type-wise, 2 chances of being positive, 2 chances of being negative, so that's a 50-50 chance. The rest of worksheet 3, you are on your own. Good luck, and let me know if you have any questions.